In this case, it could prevent the normal growth of other microorganisms if you have a certain microbe that is present from very early on that forms a biofilm. So what does this do to gastrointestinal tract development? Well, one study that was done in neonatal mice looked at this, where 100 milligrams per kilogram per day of Clomoxyl, that's a lot like amoxicillin, was given to these animals again, versus a saline control. All the bacteria in the Clomoxyl group were significantly reduced in the gastrointestinal tract, especially lactobacillus in the colon. Then they did some gene expression studies using this Affymetrix gene expression microarray technique. They found that 10 to 30% of the genes undergoing maturational changes showed modulation by the antibiotic, so that their normal pattern of maturation was either accelerated or slowed down. The MHC genes, those involved with the immune system, were markedly affected, and these are required for tolerization of luminal antigens. Okay, that's an animal study. Now, at Duke University, Dr. Cotton recently did some work, and this was published in the Pediatric Research as an abstract last year and was presented at the meetings last year. And what they did was they took a large cohort from the neonatal network of premature babies, and they analyzed data about the first antibiotic course from these babies admitted between 1999 to 2001. They identified factors associated with prolonged antibiotic therapy, and then they used some statistical techniques to look at the association between this antibiotic therapy and necrotizing enterocolitis. They had looked at 4,034 babies, and those are the ones that met the study criteria. What they found was that prolonged empirical therapy was associated with increased odds of death and necrotizing enterocolitis or death, with a trend toward increased NEC alone. And then the duration of therapy was also associated with increased necrotizing enterocolitis. Now remember, this is the use of intravenous ampicillin and gentamicin for prolonged periods of time. There are some previous studies showing that enteral administration of aminoglycosides might actually prevent necrotizing enterocolitis, and we're still trying to figure out what the difference here is. But so we're seeing that manipulation of the microbes in the GI tract might actually increase necrotizing enterocolitis if you do it one way, or decrease necrotizing enterocolitis if it's done another way. Now, what about probiotics? Probiotics and necrotizing enterocolitis. There's studies that have been done in animals, and one of the first studies that was done in animals was by Dr. Kaplan and colleagues at Northwestern University. This is a study where they take small animals, small rats, baby rats, and put them into a little plastic bag and asphyxiate them for a period of time, and then put them in a refrigerator. And I don't really know if what they are seeing in this animal model is true necrotizing enterocolitis as we see in premature babies, but it certainly is a gut injury model. And they found that with giving Bifidobacter, they could decrease the gut injury in this animal model. One of the first studies actually comes from here in humans, Dr. Hoyos and colleagues. They found a reduced incidence of necrotizing enterocolitis that was associated with administration of probiotics. That was a study that involved giving probiotics during one period of time and then comparing it to a historical control. So at the time that this study was done, there were no randomized controlled studies, and this study actually begged for randomized controlled studies. Well, now we have a couple of these randomized control studies. This is from Dr. Lin and colleagues. This is a study that was done in Taiwan. And here we have the probiotic group, the study group, 180 babies, control group, 187 babies. And if you look here at all of the results, they all favor the study group, in other words, the probiotic group, did better. They had less death. They had less necrotizing enterocolitis. Another randomized controlled study, also recently reported, this is from Dr. Kathy Hammerman's group in Jerusalem, Israel. And again, they had 72 babies and 73 babies in the control group, 72 in the study group. And here are the outcomes that they looked at. Again, all of the outcomes, including necrotizing enterocolitis. Well, the necrotizing enterocolitis certainly was favored or was decreased in the study group. Okay, so again, probiotics seem to be decreasing necrotizing enterocolitis. So it makes us very excited about possibly using probiotics in premature babies to prevent necrotizing enterocolitis. So several studies suggest benefit. Question is, should we begin to use these routinely? Is anybody here using probiotics routinely yet in premature babies? Okay, one. Okay, there are a few people using probiotics routinely in premature babies. Now, let's ask whether establishment of probiotic bacteria, which may remain with us for a lifetime, are safe, long-term, and not just short-term. Well, one of the things that's being debated right now in the U.S. is that whether we should be doing a multi-center, very large clinical trial of probiotics before we start to use this routinely. And we will probably be starting a large multi-center trial in the near future. However, there are several things that we're trying to do first to look at this. For example, if we do this multi-center trial, we may be able to establish short-term safety, but we have to have follow-up of these babies to be able to determine whether this is whether you have long-term safety. Also, there are several different kinds of probiotics. Let's say in a large study like this, you use one type of probiotic. Let's say that it is Bifidobacter, okay? And let's say that you get negative results. Does that mean that LGG or Lactobacillus or some of the other probiotic species will not have an effect? So there's a lot of logistical issues that have to be looked at before a trial like this is done and done correctly. We do know that there are some risk factors that have been seen in premature babies. And if you look over here, this is from a review article by Boyle, published in AJCN in 2005. And in immune-compromised individuals, and they consider premature babies immune-compromised, there could be a problem with probiotic sepsis. Now, in the studies that have been done so far, there have been several studies done so far with giving probiotics, and none of them have shown a baby with probiotic sepsis. However, we may not have the right culture techniques and the right studies. We don't know if the right studies have been done to actually look at the blood cultures in these babies. Okay, those may be not. Some of those might be non-culturable bacteria. Okay, so it's certainly something that we need to keep in mind, that this is one potential problem with giving probiotics. You might end up actually causing that problem, but there are other problems as well. But now there's been a lot of discussion about whether we should actually stay away from using live agents in these babies. Should we be using bacteria that have been inactivated by heat or by UV irradiation? Should we be using pre- or postbiotics? Prebiotics are agents such as oligofructose, inulin, that can be broken down by certain microbes and utilized so that those certain microbes, the probiotic types of microbes, can actually expand and thrive in the gastrointestinal tract. And then postbiotics are the things that are produced by microbes by fermentative action, such as butyrate. Butyrate is a 4-carbon short-chain fatty acid that has major effects on the gastrointestinal tract, and one of those effects is actually decreasing leakiness of the gastrointestinal tract. So that's one of the other areas. Toll receptor agonists, giving small doses of LPS, lipoteichoic acid, flagellin, or CpG nucleotides. These are agents that come from microbes that bind to these toll-like receptors. Small doses of these agents actually can prevent damage from large microbes. Okay, so it seems like it's like this is sort of a contradiction